Yes, sir. You can go ahead, sir.
Okay, so good afternoon to all, and thanks for the uh, putting my session here in the FDP, and thanks Purna for recommending my name here. Uh, my topic is molecular dynamic simulation and its application in drug discovery and development. Before I want to proceed, I just want to ask the participants about the MD simulation. So how much you know about this MD simulation? Do you have the idea of this simulation, and you already performed the simulation, or you are still means trying to perform the simulation? So it is important for me to understand the what kind of audience is there, so it will be easy uh, to focus on the topic then. So you can reply in the chat box also. Okay. Mhm. So no one has performed, or some know, some of the participants know about the MD simulation, or they already perform the simulation. Okay, yeah, I'm getting some responses. Yeah, thanks. Okay, other participants. Okay, then I can start then. The I will divide the topic like first I will tell about the basics of this MD simulation, and then I will start on or give some examples that we perform the MD simulation. Okay. Okay. Thanks for the response. So basically, the idea about molecular modeling is like we have the objects like the drug molecules, or we have a protein, or we may have nanoparticles or nanosheets or any nanostructures. And all these are, we know about these things, but we cannot see these things. And when we apply some physical laws to these atomic structures, and to find out the type of interactions between these atoms and molecules using some sort of mathematical models. And the goal, of course, is to get some geometry, the spectra or thermodynamics, or some energies or rate constant, that we recall this information. So the molecular modeling is basically the extracting this information from these structures, these nanostructures, which we cannot see. And so this is a really a microstructures, and we transform this information to a macroscopic way, so we can see these things. So there are, and we apply these physical laws to uh, do this thing. So this is what we can say about molecular mechanics. So so the basics of molecular dynamics start with the particles, the atomic particles, and these atomic particles are submitted to some potential. So some sort of potential is applied to this atomic potential is applied like U, and this expression of U is is can be expressed in terms of quantum mechanics, or it can be expressed in terms of classical mechanics. So what is difference between quantum mechanics and classical mechanics is again because quantum mechanics is again a big field, and the classical mechanics, or classical MD, is another field. So we will focus on classical MD, or classical molecular dynamics. and not and we will not discuss in much detail about quantum mechanics, but I just want to uh, put these two things here, like this potential, we can express this potential either in terms of quantum mechanics or either we can express in terms of classical mechanics. So the basic difference is the force, that potential is expressed in terms of force field in classical mechanics, while in quantum mechanics we use Schrodinger's equation to solve this and solve this force field. Sorry, uh, to solve this expression of the forces between these atomic particles. So in molecular mechanics, the basic idea is the all atoms are moving, and the forces between these atoms is a complicated function of time. So when the atoms are moving randomly, so we cannot means it is very random thing, and when we apply the Newton's law to this type of system, it is quite difficult to take the extract of the information, because uh, and this and to solve this atoms by applying some sort of pressure and temperature condition, we move the atoms little bit and then update the position and velocities uh, for each atom. So what happens is like, so it's like a minimization, but when we start at this point, so we gradually apply the forces and then we try to minimize the model. So once the model is minimized, we come at a stable state, and once we reach at the saddle point or stable point, then we can express the forces between the two different molecules. So that is what the basic idea uh, about this molecular mechanics. So we can do this either by quantum mechanics or classical molecular mechanics. So in quantum mechanics, electronic structure is a basic thing, and it is expressed in terms of Schrodinger equation, and the quantum mechanics models are quite accurate, but are also expensive. Expensive is in the terms of computational time, so it takes a lot of time, computational time. So even if you want to calculate the this if you want to do the quantum mechanics on supercomputers also, it takes a time. But if we go for the classical molecular mechanics, these are empirical forces. We calculate the empirical forces, and as they are empirical, so they are less accurate, but quite a fast. So it is possible to apply these empirical forces for the protein molecules, because proteins are quite big one, and we cannot solve the protein structures using quantum mechanics. But we can do for the small molecules, but not possible for the big protein structures. So for that, we need molecular mechanics. There is an approach of mixed quantum classical mechanics also. So recently some papers are also coming for this mixed quantum classical mechanics uh, based simulations also. So what are the things important before we start with these molecular simulations? So we need to express a degree of freedom of atoms. So for that we need basic coordinates, which we get from the PDB. Then we need to set a boundary condition. So periodic boundary condition we need to set. Then we need to uh, calculate the forces, interaction forces between the atoms, and then this can be done using applying some sort of force fields. And then we need to generate the configuration of atoms. So we need to apply some mathematical equation to solve this interaction. So this is a typical PDB file. So which you can get from Protein Data Bank. So this file, if you visualize this file in a normal text editor like Notepad or VI or Vim editor, then you can see like there is a information. So the type of atoms are given, then atomic numbers, then atom types, then type of residues like amino acid residues, or is there any heteroatom is there that is given, then the chain numbers are given, and then residue numbers, and then these are the three coordinates, so x, y, z coordinates of these different atoms. So this file is called as the initial coordinates of this protein structure, and then we submit these initial coordinates for molecular dynamics. Uh, so before submitting, we need to take account of different things. So first we take these coordinates, then we apply some force field. So we need to choose the force field. Now, depend upon the force field, because force field are given for is assigned with the terms. So sometimes it is difficult to identify some heteroatoms if it is there in the PDB. So we need to create the force field for that unknown residues, because most protein residues are known. So force field are basically already built for the amino acid molecules. So if the amino acid residue is there, it is easily identified directly. So we don't have to rename the amino acids. But if we have some heteroatom in the PDB structure, or if we perform a docking study with some uh, inhibitor compounds. So these inhibitor compounds are not known for the force field. So we need to create the force fields for that heteroatom. It can be done as per the program which you are going to use and which type of force field you want to use. So there are different kinds of force field. I will show you shortly these different force fields. So you can choose any of these force field to create the model. Once the model is created, then we need to think about how much is the time step we need to take. So if we compare this molecular description, so normally if we talk about the quantum mechanics, so it takes so the motion, electronic motion within the atoms, within the molecule, are in the range of femtosecond, picosecond, and up to nanosecond. So some changes, when we want to observe some changes in the molecule, that changes last up to some few nanoseconds. So if we perform a few iterations, like 100 iterations or something like that, then we can get the information about the electronic structure of that uh, compound using quantum mechanics. But for classical mechanics, it is quite challenging to know within the nanoseconds. So because this changes happens from few femtoseconds to up to seconds. So the time interval required for these changes are having a big time frame is required, and that's why the longer the simulations are always a better one. So previously the simulations were like few nanoseconds, 1 nanosecond or 10 nanosecond simulation was also enough previously, because at that time computational resources were not that good. But now with this revolution in the computational sciences, we can now perform a simulation from few microseconds to milliseconds to seconds also. So if you perform a simulation for few seconds, then it is always good, but you need to think about the time required and how much resources you are having with you for performing the longer simulation. And also depends upon what type of interaction you want to get from that simulation. So if it is drug and small molecule interaction, then you can go up to some 500 nanoseconds or 1 microsecond, and that is could be enough. And because it is not like one molecule you want to simulate, but some like 10 molecules or 15 molecules you want to screen, and in that case, uh, even this 100 nanosecond simulations also will be fine then. Now, why and how to assign the time scale? So when we uh, define the time step in simulation, we need to think about the different changes happening inside the molecule. So if we consider the bond stretching, so normally the time scale of bond stretching is like 10 raised to minus 14 to 10 raised to minus 13. So this is the quickest one. And then elastic vibrations, then rotation of surface side chain, then hinge bending, then allostatic transition, local denaturation. All these are taking place in a in few seconds to up to like nanosec femtoseconds. So when we assign when we calculate the forces in few femtoseconds, so we can know about the bond stretching. But but to understand the different interaction or denaturation, then we need to perform this simulation for longer time. So the longer is the simulation, then more is the information we get. But the we need to understand the small forces also. So that's why normally the 2 femtosecond time step is quite enough because it covers the bond stretching motions. So dynamic simulations are limited by the highest frequency vibration. So this is kind of the highest frequency vibration. And if we, and that's why we have to limit the speed of the simulation, like few femtoseconds, and we cannot go beyond then. We don't have to go beyond, but we need to set up to this one to understand this stretching vibrations, up to stretching vibrations. So we can get all the information between the interactions. And then the force fields are expressed in different terms like stretching frequency, then bending, the torsional angle, the van der Waal interaction, electrostatic interaction, and then cross-sectional forces. So this could be like surface tension or like that. So these interactions are important, and the force fields are like these are like scales or ruler. So when we have a scale, so we know that we are allowing a movement of atom, but also we set the limit for the movement. So it's not like the bond is stretched. So when bond is started stretching, so if it is like oxygen and carbon is bonded, and if we want to stretch that bond, so it's not like it is going on stretching and stretching, but it will be set into the limit. So every movement will be limited as per the scale. So the force will define that scale, and that's why we are not creating the hypothetical case, but it's a practical model. Then it's more practical that this much the angle could bend and not not beyond that limit. So force field also sets the limit for that motions. So and they're quite important to understand the motions between the atoms. So if we see the force field, they are different for the protein, small molecules, and lipids. So if we go for the protein, then Umber force field is quite good force field, and most papers which are published with this with these molecular dynamics simulations are with these Umber force field. Currently, Umber 14 force field is in force, and recently Umber 18 force field also introduced. Then charm force field is another one. It's currently the charm has charm 36 force field. Then Gromos Ophilis all atom force field. Then general umber force field, GAF that is called. And this GAF is normally for the small organic molecules. Then Mark molecular force field, and then OPLS3, and OPLS3 recently introduced by Desmond. Then lipids can be defined by different force field. So there are S lipid force field in Gromax, and umber lipid force field is also available there. So we need to define the force field. Apart from that, the DNA and RNA are also defined by the force field. So umber has a separate force field for the DNA and RNA. So if your molecule or if your protein is interacting with DNA chain, then you need to define that RNA or DNA using that force field. Then the next thing is water, because simulation performed without water cannot give us the real understanding, because you know that our body is full of water. Even the all living organisms are having the water there, and so it is impossible to uh, consider the simulation without water. So, and it's not just the water, but also the salts and ions. And that's why when we start with the molecular dynamic simulation, we need to solvate the structure, or we immerse the structure inside the water, or we create a PVC with the water. So the protein is completely immersed inside the water. So hydration is there, and then we also neutralize the protein structure by adding some counter ions like sodium or chloride ions. Sometimes we also, if needed, we also set lipids. So we can, like if you want to study some membrane proteins, then in that case, the lipids are built, and then the protein is attached or inserted between the lipids, and then we perform the simulation. So this type of, if you take water, then it is more explicit. So there are two types how we can account the water. So the water can be considered as explicitly or implicitly. So in explicit condition, it's like we have a water models like tip 3, tip 3p, or tip 4, or SPC water models. But when we have the water, then the water molecules are quite large. So they are not large as a size, but we need to take a lot of water molecules to immerse one protein inside, and that creates a very big system. And the bigger the system you take, you need a longer time for the simulation. So it comes with a computational cost then. And the second thing is, we can also set water using implicit solvent solvation models. Like there are like MMGPSA or PBSA solvation models are available. We can use that solvation model. So we don't, we are not putting the water, but we are using the mathematical models to calculate the forces exerted between the protein and ligands. And then here these explicit solvation models are again defined in terms of SBC or PBC. So SBC is a stochastic boundary condition where we hydrated the protein, particular region of the protein, and we apply some forces. So water will stay there only in that particular region. And this is more complicated to perform, but if we do this, then we don't have to put much time for the simulation, so your simulation time will be reduced. But what more useful is the PBC. So when we have a PBC, so in case of PBC, we we immerse the protein inside, and the whole protein is actually immersed inside the water molecules. So water is all around there. So this is a kind of explicit solvation model. Then implicit solvation models can be defined by Poisson-Boltzmann equation or generalized Bond equation. So when we have an implicit solvent model, then it can be defined in terms of Poisson-Boltzmann model. So it is called PBSA solvation model. And if we have a generalized Bond equation, then it is called MMGBSA solvation model. So these are quite famous solvation models, which are normally used for in the simulation. Then once everything is set, we need to set temperature condition, pressures, then the other external forces. So the important thing is periodic boundary condition. So what is periodic boundary condition? So if we simulate the protein as a whole, then what will happen is some residues on the protein will be on the surface and some residue are maybe inside the core. So force exerted on the surface atoms will be different, and then the force which is there in the inside the that core part will be different. And to avoid this issue, we create a PBC. So when we create a PBC, so one PBC is like the center atom. So your atoms are inside here, and then it is surrounded by the second structure. So water can freely move, and even your structure is going to move from one side. So when from right hand side to left side, so if it start moving, so it can go, and there will be no change. So and then the forces which is exerted will be constant then if we use the periodic boundary condition. So this is how we can avoid the surface artifacts in simulation. Then we need to define the ensembles. So there are different types of ensembles we can define, like NVE, that is called when we have Newtonian dynamics. So there are like Worlet scheme or leapfrog or velocity Worlet or gear type ensembles. So these are called micro-canonical ensembles or NVE. Then there are canonical ensembles like NVT, where we can have indefinite source of temperature. So once the temperature is dropped, then we can take the temperature. So your protein model will take more temperature from the source. So we have a source of temperature, so it will not deplete. And if we have this NPT ensemble, so it is called isothermic isobaric ensemble. So there will be a fixed pressure and the temperature, infinite source of temperature. So when the temperature is increased or decreased, there will be equilibrium. Equilibrium will be reached between these two states, and then the pressure is also fixed. So we set a pressure and we set a temperature here, and so this is what the basic ensemble which is normally used for production run MD simulation. While in case of equilibrium simulation, we normally use NVT simulation. Then the time scale, as we discussed in some previous slides, that how much we should define the time step. So normally the simulation require a short time step, like few femtoseconds. So normally 2 femtosecond is considered as a good, because it also good in terms of computational time. So we need to consider the computational time also, and so the movement between the atoms. So it is, and then as the structural changes are from few nanoseconds to microseconds to milliseconds or longer, so we can define that. And then previously the simulations were not parallel, but now with the GPU enabled simulation, we define this simulation box in terms of different cores. So you can, when you perform the simulation, the actual system is divided and it causes the parallelization. So more parallel the system, so we can do longer simulations if we divide this uh, system into or if we parallelize the system. So now there are like different GPU cores are available, or the CPU chips are some modern CPU chips are also available, or NVIDIA graphics card like GTX graphics card is rolled out. So they are quite fast doing the in performing this type of simulation. Or even the Anton chip, which is designed by uh, Shaw Research, is also very good uh, for performing the this GPU enabled simulations. And then if we summarize this MD simulation, so we need to select the interaction model. Then we need to select the boundary condition. Then we need to set initial conditions. So we need to set initial condition in terms of PDB. So position and velocities we will get, and then selecting the ensemble. So which ensemble you want, and then selection of target temperature, density, pressure, selection of integer, thermostat, which type of thermostat you want to choose, barostat, and then perform the simulation until equilibrium is reached, and then running the production simulation, and then analyzing the simulation. So this is how we can go from uh, preparing the system to performing simulation and analyzing the simulation. And there are different types of software programs are available like Charm, Umber, Bromos, Bromax, and NAMD packages are available, or even a Desmond from the Schrodinger or is also available to perform the MD simulation. So using these software programs, we can perform MD simulation, and then we can get the information about the structural. We can get a structural data, or we can get energy data, or some properties from this simulation. So, so the next point, I will take some examples uh, about this molecular dynamic simulation, which we have performed in our lab. But before proceeding, if you have any questions, then we can have a small question and answer session here, and then I will start moving then. Is that okay?
Yes, sir. Good afternoon, sir.
So, so we take a small pause, and then again we will start for the next part. But if someone has questions on this part, then we can take questions, and then we'll move towards the next step.
Sure, sir. Sure, sir. Any questions from participants?
If you have any question, you can also use chat box to and write your question there.
Any queries related to topic from the participants? You may directly talk with the speaker.
Yeah, I think there is one question.
Thank you, sir. Carry forwards.
Okay, no question.
Yes.
Okay, should I start moving then?
Yes, sir. Sure, sir. Start your presentation, sir. Thank you, sir.
So I will take one example where we perform molecular dynamic simulation on this SARS-CoV Mpro. So you know about the Mpro. Is Ganesh, any question? Okay, no. So so you heard about this Mpro is a main protease which is responsible for cleavage of the nucleic acid core, and after cleavage the RNA is released. RNA of the SARS-CoV is then released inside the human host. So to have the active RNA released, the this viron need to cleave this protein nucleic acid, and to cleave that it utilizes this protease enzyme. And as the structure of this protease was quite available earlier in Feb, so you can see that there are a lot of papers were there on the M protease. And we also choose this M protease. So we taken the 6LU7 PDB ID, and we did a molecular dynamic simulation. So before we are going to proceed on molecular dynamic simulation, what we did is we first taken the structure of this M protease, and then we compare the sequence of this M protease with the uh, previous infection caused by SARS-CoV-1 or SARS-CoV viron, and also compare this sequence with the MERS-CoV. So after comparing, we observe that the sequence is almost 96% identical. So there is some variations in amino acids, but the variation is not that big. So 90%, 90 to 100% similarity is, like, we can say, like it's quite similar structure. The sequence was quite homologous, and that's the reason the structure is also available quite fast, because it is easy for the phasing thing. So when the structure was available, it was the 6LU7 is the first structure reported, and it is with the N3 inhibitor, which is covalently bound there. So our target was to uh, have the inhibitor either covalent or non-covalent inhibitor. So as a covalent inhibitors, we taken some active library. So it's a covalent focus library. We taken this SNX covalent focus library, and for non-covalent interaction, we choose the FDA approved inhibitors, which are already there in the market. So, and then we perform the screening. So after screening these covalent and non-covalent inhibitors, so non-covalent inhibitor was bound to this cavity there, which is you can see here. And for the covalent interaction, the inhibitor need to form the covalent bond with the cysteine residue. So cysteine 145 is a key residue for that covalent interaction. So we perform the this covalent docking. So we use Schrodinger to do this screening. So covalent as well as non-covalent screening. And then after screening, we got the structure was already there for the 6LU7, and then we dock the different ligands. So they are either covalent or non-covalent. And then we calculated the binding free energy, because docking score was quite random. So docking score, we are not relying much on the docking score, but instead we calculated the binding affinity using this uh, the solvation model we discussed. Like GBSA solvation model we use, MMGBSA model, and then calculate the binding free energy. Once we got the binding free energy, then we rank the molecules as per their binding free energy, and those molecules which are having a least energy, or the most minus energy is chosen for the next phase. So the next phase was to prepare the model for MD simulation. So when you want to prepare the model, then you need to prepare the system. So system is prepared first by assigning the force field. So we use umber force field, umber 14 force field for protein, and for non-covalent interaction and also for the covalent interaction we use GAF force field. So it's a general atom force field from the amber using anti-chamber package. So we define the different bonds even for the cysteine and modified cysteine and this small molecule. And once we define these parameters for these force field in a leaf editor, we then solvated the system using periodic boundary condition and uh, uh, as a cubic box, and then we neutralize the system using appropriate sodium and chloride ions. Once the system is neutralized, we prepare the coordinates file and topology file for the amber, and then we started the minimization. Uh, Now, when we start the MD simulation, the simulation need to be in a different phases. So the first phase of simulation should be like minimization phase, then the equilibrium phase, and then production simulation. And you should not jump directly for the production simulation. The reason behind is when we have coordinates, initial coordinates, there is a lot of strain there, and the structure is normally in a higher energy state. So when you start minimizing the model, that what happens is these bonds start to move. So if there are some penalties are there, if there are some steric clashes there, so these steric clashes start to go away. So when we start at the first stage, so we need to minimize the system. So there are different kinds of minimization protocols. One protocol is Brownian minimization, where uh, we use NPT ensemble and minimize the model. So energy minimization can be done by Brownian minimization. The second option is Stiffness gradient method of minimization. The reason of using the steepest gradient method is, and the advantage of steepest gradient method is when you start minimizing. So it's not like we always get a minima, because the structure of the protein, this minimization thing is very random. So maybe I can use, there is no board option. So if you have the system at a particular energy level, so when you start minimization, the energy will drop, and then it might be possible that you may get another high energy state. So it is very random thing. So you may get a minima and then again a maxima and then minima and maxima. So these things, so it is quite a zigzag thing. So you cannot predict accurately that after 1,000 step of minimization or after 2,000 step of minimization, you will reach a minimum step. But sometimes you may reach at the maxima also. So to avoid this, steepest gradient method is used. And in steepest gradient, what happens is, though you define 1,000 step of minimization or 2,000 step or 5,000 step of minimization, after defining the steps, your model start minimizing. And then when the minimization reach, then though it is in the 100 step or 1,000 step or after 2,000 step, if your model is now minimized and there is no chance of further minimization, then it stops. And then you get the protein structure which is minimized actually, and not comes at some saddle point or at high energy state. So that is the advantage of using the steepest gradient method. Once you minimize the system, then you need to equilibrate the system, because in minimization normally it's a very gentle way to shake the structure. and and then when we do the MD simulation, it is not that gentle, but there are a lot of forces we applied. So if we start directly the production simulation or MD simulation, then what will happen is like the protein, the structure of the protein may collapse. So there may be possibility that the your model is not properly equilibrated, and then you will get a very zigzag RMSD. So to avoid that, you need to equilibrate the system. So equilibration, in equilibration condition, we normally shake the structure quite a lot using Langevin dynamics or shake algorithm. So what happens is like your structure is quite heavily uh, uh, forced to accommodate the minima conformer, and then we start achieving the temperature which is you utilize for the MD production simulation. So like if you have a 300 Kelvin simulation, so you can have a restraints on the solvents, or you can put a restraints on some atoms so that they will not move in the equilibrium state, and then gradually you allow them to release. So when you start a production simulation, your whole protein is now simulating, and not you are not putting some restraints on your atoms. So for that you need to equilibrate the model, and once the model is equilibrated, then you can start the production simulation. So this is so in this three step you can do the MD simulation. Once you start the simulating, then the question arises like how much time we should simulate the system. So it depends normally on the how much computational resources you are having. So if you're having good computational resources, then 100 nanoseconds, 200 nanoseconds, or 250 or 500 nanosecond simulation is quite good. Uh, but if you don't have that much resources, then you can limit the simulation for 10 nanoseconds or 50 or 25 nanoseconds. And then if you get any good structure, then you can replicate that simulation again to see whether that interaction is stable. So normally when we start the simulation, we observe the RMSD. So I can show you some information here. So I will stop sharing here, and then I will share my screen. So, so if you see, can you? Okay, so if you see here, this is the RMSD, and this is the time. Can you see this another window?
Oh, yes, sir. It is visible.
Okay. Okay. So if you see here, so you can see like at the initial stages, there is uh, RMSD is not stable. So the energy is increased somewhat up to this 25 nanoseconds. And you can see that up to 25 nanoseconds, the trajectories are stabilized. So at the end of this simulation, you can see that all the simulations are stabilized, and the RMSD is at the stable state. So and then you can because this is very needed, because if your system is not stable, then you cannot calculate the binding affinity, because once your model is stable, then only you can calculate the interaction between the molecules. So this is the good state to calculate these interactions. So if you calculate these interaction at these stages, then you will get a very good information about your simulation. So this is how we need to focus on that. So here you can see this molecules which is having a non-covalent interaction between the cysteine and this the site of this M pro, and you can show you some more. And here, these molecules which we identified through this simulation, where they are having a stable interaction with these. And once the interaction stabilizes, we calculate the energies between these molecules. So how they are interacting and different non-bonding interactions which were there between these molecules. Okay, then I will continue on my slides. So this is one example we did uh, MD simulation, and then another case where we perform the MD simulation is about the DNA gyrase. Okay, so the target, our this is another paper we recently published, and in this case, our aim was to in develop some DNA guidance and topoisomerase inhibitors. So our collaborators work on the synthesis, and they perform the inhibition assays as antimicrobial compounds, and the compounds were mimicking the coumarin and uh, the fluoroquinolones. So our target is to find out the inhibitors that could be DNA gyrase or topoisomerase. So if we see this DNA gyrase, then inhibitors could bind to the B subunit of DNA gyrase, or it can bind to A subunit of DNA gyrase. So this structure is A subunit. The A subunit has again three different sites. So these two sites are for fluoroquinolones, and there is one new site. It's called NBTI site, or novel bacterial topoisomerase inhibitor site. So it's NBTI site, and this is the third site which is there on DNA gyrase. So when we started docking study, so we docked our molecules here on DNA gyrase B subunit. Also, we performed the docking on cyclophosphamide binding site, or most specifically fluoroquinolone binding site, and also we perform the docking studies on NBTI site. Now, when we do the docking, uh, we got some good poses and some bad poses also, and at that time it is and even and after docking, it is quite difficult to predict that this compound is good or this compound is bad, or this is good interaction and this is bad interaction. And that's why it's we need to perform MD simulation. So when we did MD simulation, we come to know about that some interactions are good and some molecules are really good interactors, while some molecules are not that good, and also about the choosing the right site. So when we perform the simulation on the DNA gyrase B subunit, then we observe that molecules were not able to bind to the in the exact way where this novoiosine or coumarin nucleus binds, and we concluded that this is not the good site for our compounds. So we didn't get very good interactions there. We also compare our binding studies on the NBTI site, and we also observe here that our compounds were not good inhibitors at this site. But when we studied the docking and then MD simulation and binding energy prediction, we observed that our compounds were binding quite good uh, at this fluoroquinolone site. So to come to that conclusion, we need to perform some simulation. So here we calculate the interaction. So this is how our molecules were interacting at fluoroquinolone site, and this is how the molecules are interacting at no biosine site. So what we observed is at no biosine site, the compounds were not able to get inside the pocket, which is a very narrow one and long and narrow pocket adjacent to ATP binding site. And as our structures were more flat, So they are not able to get inside that narrow binding pocket. So that's why they are not able to bind very efficiently at the no voisin site. We can show you some information about that case. So this is what we perform. So when we start the MD simulation for that for these compounds, so first we run a small 100 picosecond Brownian dynamics at 10 Kelvin using NVT ensemble, and then we restrain the heavy atoms of the protein with some force. So there is a restraint, and then we perform the Langevin dynamics with NVT ensemble using Burson thermostat, and relaxation time was like 0.1 picosecond, and time step is 1 femtosecond. And then we perform 12 picosecond Langevin dynamics uh, at 10 Kelvin and 300 from 10 Kelvin, and then again at 300 Kelvin at 1 atmospheric pressure. So we use NPT ensemble because we need to transition. We need to move from the minimization step to production simulation. So the minimization step is like 10 is at temperature 10 Kelvin, and our production simulation supposed to be at 300 Kelvin and 1 atmospheric pressure, and there is no restraint. And when we start minimization uh, or at the first equilibrium state, it was like there was some restraint. So we need to remove the restraints gradually. So when we start moving, so we need to start gradually from minimization to production simulation. So gradually you can see that we reach to the 300 Kelvin temperature, and then we set that ensemble to NPT, and then pressure condition to 1 atmospheric pressure, which is exactly same as that of the production simulation. But this condition we set at the last stages. So what happens is like we perform this with some restraints and then without restraints. So this allows us a smooth switch from the minimization step to production simulation, and that is important in MD simulation studies. And then we run a general protocol of MD simulation, which is already there in the paper. Then you can see that if you see the trajectories, so when we calculated the trajectories, then we observed that after 10 nanoseconds, there is a good stabilization of the RMSD. So RMSD is quite stable. Here you can, here this is DNA guided A subunit, and the site is, and we compare this simulation with a co-crystallized ciprofloxacin and our compound. So we observe that the trajectories are quite stable for both of the experimental compound, also with the co-crystallized structure. Then we did it for at another binding site. This was this coumarin binding site, like no voisin. So we compare the simulation with no voisin and with our compound, and we did a comparison at par E subunit of DNA guidance also, and then we compare the simulation with NBTI site. Once we compare, we also calculated the fluctuation of this amino acid fluctuation to compare to see whether the ligands are stabilizing the loop regions also. And then when we compare the binding energy, you can see the binding energy of our compound was quite similar to that of the binding energy of co-crystallized structure. So this co-crystallized structure is actually the structure of ciprofloxacin. So when we compare the binding energy of our compounds with ciprofloxacin, we observe the similar trend, and that's how we conclude that our compounds could shown the activity of and as antimicrobial compound. because they are interacting in a quite similar way, the way in which the ciprofloxacin interacts with the DNA guide A subunit. But when we, but this is not the case when we compare it with the with another docking site. So here it's like when we compare the binding energy with DNA guidase B subunit, which is actually the no bias in binding site or common in binding site. So here you can see that co-crystallized structure is more stable. So interaction is more stable with the co-crystallized structure. But when we study the interaction of our compounds were not able to bind or their binding efficiency is not that good. So they were not good inhibitors at this site. And we also compare uh, with the with the with the topoisomerase 4 par E subunit, and we observe the similar trend that our compounds were not good inhibitors at this site. And then we also compare at another binding site, which is the NBTI site. So it's a novel bacterial topoisomerase inhibitor site of DNA guidase A subunit. And when we simulate and calculated the binding energy of our compound was this much, which is very low as compared to the binding energy of co-crystallized structure. And this is how we concluded that our compounds were not binding to NBTI site or not they are binding to DNA guidase B subunit, but they are interacting with fluoroquinolone site. And energy is also good in that simulation. So that conclusion we come from this observation. Okay. So this is how we can apply the molecular dynamic simulation for drug screening. So to do that, we need to have a dock structure of your ligand. So you can use any docking program, and then you have to select the least energy structure, the energy which is minimum, and then that structure you can then submit for the simulation. So uh, you can use any of the program, like Gromex is freely available tool for performing MD simulation. So if you have good computational resources, or even on a desktop with some course, CPU course, you can do that simulation. Or you can use some commercial package like Schrodinger or Amber also to do that simulation. Then it's not only the drug discovery where you can use the MD simulation, but also you can use MD simulation for drug development. So drug development aspect of the MD simulation is different as compared to the drug discovery aspect. So in drug development, what is needed is we need to have a system, or we need to have our compounds which will we need to calculate the interaction. So here I will show you one example. In this is our ongoing study where we are determining the solubility uh, of the protein, sorry, not protein, the polymer and drug for the amorphous solids. So you know that number of drugs are not soluble in water, and these drugs which are least soluble in water, and there are some good candidates like some like ibuprofen like drugs which are very poorly soluble. Or to enhance that activity of these drugs, we need to enhance their solubility. So amorphous solid dispersions is a good choice for that, but choice of polymer is very critical factor. So in this simulation we perform a simulation using a polyvinyl pyrrolidine polymer. So PVP polymer we choose, and then we added the drug. So when we start adding the drug, so here you can see that this this globular part is actually a polymer, and this blue color structures are actually the drug molecules. So this is the one simulation where we taken a drug and then we observe the interaction. And then we calculated the hydrogen bonding interaction, then solubility parameter and cohesive energy of these polymer and drug. And we observed that at 30% concentration, there is good interaction of polymer and the drug, and hydrogen bonding between the drug polymer is more as compared to drug. So when drugs start to interact with each other, so other drug molecules, so the interaction is quite tight, and that makes it more crystalline. So as the drug becomes more crystalline, so it will the solubility of that drug decreases. So the objective of this amorphous solid dispersion is to have the polymer which will have more interaction with the drug, and there will be a less interaction with the drug and the other drug. So we concluded that when we have a system with 30% polymer and 30% drug and 70% polymer, the interaction is higher with the polymer. But when we have opposite case where we have around 70% drug and 30% polymer concentration, then drug-drug interaction uh, are more. So there is more hydrogen bonding between the two drug molecules, and hence the interaction between the polymer is diminished, and the solubility of this system decreases. So this is how we can apply the molecular dynamics simulation to study different state. Even you can determine the interaction of nanoparticles with the membranes, or the solubility parameter determination using MD simulation. So there are like a large number of applications of these MD simulation studies. So here I can summarize that we are having these computational resources from CSC. So CSC is the centralized system for supercomputer resources in Finland. So currently we are having the supercomputer, which is having a speed of around 1.5 petaflops. And then in this year, in 2020, we got another supercomputer, Mahati, which is having a speed of around 7.7 petaflop. And then next year, this Luby supercomputer is going to be rolled out, which is having, which will have a speed of more than 200 petaflop. So currently, the Finland is quite leading in the supercomputer resources for performing the simulation, and also quite a long simulations. So availability of these resources is very key for performing MD simulation. We also recently on 31st December, we published our review. So if you go through this review, then you can also understand in a very detail about this molecular dynamics and its applications. Also, the review also gives a very detail about the drug simulation and also the molecular dynamics simulation in pharmaceutical development. So there were quite good examples were given, and there are some like around 550 references are there. So you can cross-refer that references, and you can study this. You can go through this paper. So this paper is published along with the top scientists in the field, like Alexander Bonvin, who is the founder of Haddock, and Rodrigo Honrato, and Andre Zufor, Alexei Krabatev, which is they are very key persons who are doing the MD simulation of nanoparticles from Uppsala University, and there are like quite a good collaboration we have on these aspects. So if you go through this review, which is openly available, so you can just need to type the name and you can see this review and you can read that review paper. So you will also get a very detailed understanding of MD simulation. Apart from the MD simulation, we also did protein crystallization study. So this is a short about our lab. So this is a nanotemper nano DSF, which is used for determining thermal stability of protein, and also for calculating the interaction, and also between the ligand and drug interaction. For that purpose, we use this nano DSF. And then this is protein purification system. This is an AICTE system for protein purification, because for X-ray crystallography we need protein crystal which is having more than 90%, 90 to 95% or more than 95% purity. So we need to purify the protein. Then once the protein is purified, then we need to set the protein into the crystallization wells. So these are sitting drop crystallization wells. So this is the mosquito which is used for setting the plate. So this is very tiny plate, so you cannot do it by hand. So you can set a different screening. So this is the protein screen for protein crystallography. So this is one screen, and you can set the 96 well plate screen for protein crystallography, and then you can detect the crystal in any of the screen. So crystal determination is also very automatic. So we have this formula test system where the crystallization wells are placed into this uh, formulatrix well, and then automatic robotic arm is scan the structure, and then it takes the photographs. And once we got the crystal in any of the well, then that is an analyze whether the protein crystal is are from to confirm that the crystal is from the protein and not from the solvent or any other precipitate there. So to once we confirm that the protein, the crystal structure, crystal is of the protein, then we submit the protein samples to synchrotron resources. This is one of the synchrotron resources. So we have a max4, also this diamond, or then then in a green lobe also we have this different synchrotron resources where we can ship our protein sample there and can analyze the protein structure. Once the protein structure is analyzed, is then deposited in protein data bank. So this is how the life cycle of protein data bank. So how the protein structure comes. So it starts from the small microorganisms, and then it ends with the structure of the protein, which is in the 3D coordinates that we take for the MD simulation, and then again perform the simulation. So this is our team, and this is my mentor, Professor Auti Salwayan. I'm thankful to her, and also thankful to Abo Academy University, then Academy of Finland, then Todd